After gastrulation, a remarkable transformation begins in the developing embryo. This is the process of organogenesis, where specialized tissues and organs arise from the three germ layers: ectoderm, mesoderm, and endoderm. One of the most critical events in organogenesis is neurulation, the formation of the neural tube, which will eventually give rise to the brain and spinal cord, the control centers of the nervous system. Neurulation is a carefully orchestrated process involving a complex dance of cellular movements, signaling pathways, and tissue interactions. Understanding neurulation is crucial because it lays the foundation for the entire nervous system. Errors during this critical period can lead to severe birth defects. The study of neurulation and organogenesis provides a window into the fascinating and intricate mechanisms that govern the development of all vertebrates, including humans. Neurulation begins with the formation of the neural plate, a thickened region of the ectoderm, the outermost germ layer. The ectoderm is destined to give rise to the skin and nervous system. Signaling molecules, like those from the underlying mesoderm, induce a specific region of the ectoderm to differentiate into neural tissue. This region becomes the neural plate, the precursor to the brain and spinal cord. The cells of the neural plate are called neural progenitor cells, which have the potential to develop into various types of nerve cells and supporting cells of the nervous system. The formation of the neural plate marks the first visible sign of the developing nervous system. As development progresses, the neural plate undergoes further changes. The flat plate begins to fold inward, forming a depression called the neural groove. Along the edges of the neural groove, raised ridges called neural folds form. These folds are critical for the next stage of neurulation. The formation of the neural groove and folds involves changes in cell shape and adhesion. Cells in specific regions of the neural plate contract, causing the plate to buckle inward. Changes in cell adhesion molecules help to orchestrate the movements and interactions of cells during the folding process. In a remarkable feat of biological engineering, the neural folds move towards each other and fuse at the dorsal midline of the embryo. This fusion converts the open neural groove into a closed tube, the neural tube. The neural tube is filled with fluid and becomes completely internalized within the embryo. The closure of the neural tube is a critical event in neurulation. The anterior or front end of the neural tube will develop into the brain, while the posterior or back end will form the spinal cord. The fluid-filled central cavity of the neural tube will become the ventricles of the brain and the central canal of the spinal cord. Section 5. Neural crest cells, architects of the peripheral nervous system. As the neural folds fuse, a population of cells at the crest of the folds detaches. These cells, called neural crest cells, are a remarkable example of migratory cells in the embryo. Neural crest cells migrate extensively throughout the developing embryo, giving rise to a wide array of cell types. Neural crest cells contribute to the formation of the peripheral nervous system, including the sensory neurons, autonomic neurons, and Schwann cells that myelinate peripheral nerves. They also give rise to pigment cells, some bone and cartilage in the head, and parts of the adrenal glands. The migration and differentiation of neural crest cells are essential for the development of many structures in the body. Section 6. When development goes awry, neural tube defects. The closure of the neural tube is a critical event, and errors in this process can lead to severe birth defects known as neural tube defects. These defects can affect the brain, spinal cord, or both, depending on the location and extent of the closure defect. One of the most common neural tube defects is spina bifida, which occurs when the posterior portion of the neural tube fails to close completely. This can result in varying degrees of paralysis and other disabilities, depending on the severity of the defect. Another serious neural tube defect is anencephaly, where the anterior portion of the neural tube fails to close, resulting in a baby born without parts of the brain and skull. These conditions highlight the importance of proper neural tube closure during early development. Section 7. Organogenesis, from simple tubes to complex organs. While neurulation is underway, the other germ layers are also busy forming the rest of the body. The mesoderm, lying between the ectoderm and endoderm, gives rise to muscles, bones, connective tissue, the circulatory system, and the urogenital system. The endoderm, the innermost layer, develops into the lining of the digestive tract, respiratory system, and other internal organs. Organogenesis involves a complex interplay of cell proliferation, differentiation, migration, and programmed cell death. Cells in different regions of the embryo receive specific signals that dictate their fate and behavior. For example, cells in the developing limb bud receive signals that tell them where they are along the proximal distal axis, anterior-posterior axis, and dorsal-ventral axis. Section 8. The interplay of signaling and timing. The development of organs and tissues is a highly regulated process that depends on precise timing and coordination. Cells communicate with each other through signaling molecules which act like messengers, conveying information about position, identity, and fate. These signals activate specific genes within cells, triggering a cascade of events that lead to differentiation and morphogenesis. Disruptions in signaling pathways or timing can have profound effects on development, leading to birth defects or other abnormalities. Understanding the molecular mechanisms that control organogenesis is an active area of research, with implications for understanding birth defects and developing new therapies. Section 9. Conclusion: A Symphony of Development. Neurulation and organogenesis are remarkable examples of the intricate and highly coordinated processes that govern embryonic development. From a single fertilized egg, a complex multicellular organism arises, with specialized tissues and organs working together to maintain life. Understanding these processes provides insight into the origins of our own bodies and the delicate balance of cellular events that make life possible. Through the study of developmental biology, we are beginning to unravel the mysteries of how genes, signaling pathways, and cellular behaviors interact to shape the embryo. This knowledge is not only fascinating from a scientific perspective, but also has important implications for understanding and treating human diseases. By unraveling the complexities of development, we gain a deeper appreciation for the beauty and elegance of life itself.